Hi, this is Katie, one of your Bio 110 instructors. And today I'm going to show you a little bit how to do this part 3 observation of gram-positive and gram-negative bacteria, this activity. So there's a microbiology lab PowerPoint that you're going to use, and there's 2 slides that have these images on them. So what you're going to do is draw and color a few of the bacteria cells observed in each image. And this is slide 17, 18, but it is a PDF, so they may not be exactly there, but it's going to look like this. There's 1 through 6. I believe it's 2 different slides. And let's say if we look at one, you're going to classify the bacteria according to shape and arrangement. You're going to identify the bacteria as gram-negative and positive. So if you look on here, remember if they're purple, they're gram-positive. If they're red or pink, they're gram-negative. So purple, P for purple for positive, and they stain purple with the gram stain because they have more peptidoglycan in their cell wall. So again, peptidoglycan, purple, and positive all start with P. So here's red as negative. There's pink too, but just don't think of that as P. Just think of it as a diluted red. Okay, so if you're going to look at something, here's an example here, then you want to draw it on there. Now, some people draw things on different ways. Honestly, I usually use PowerPoint, not this. Some people print these out and then color them and draw them. Other people, I don't care as long as they're done. But what I'm going to do is, if I look at this purple one here, and I went up before I was in home, and then I went to the draw tab. And then the draw tab has all these. And there's other ways you can do this. But if I want to do slide 2, then I look at it and it's like, these look like little lines all hooked together. So that's too big, that circle. So I'm going to go make it thinner. Let's see about that. And then also I'm going to make this larger, so it's easier to see for me and for you. Okay. And so here it is. Let's see, maybe a little bigger, just so we can see it better. And you don't have to draw them all, but just draw a few so we can get the idea. Okay. So there's the idea here. Maybe I'll do a few more. But the main thing you see the color, it's purple. So that's going to be gram what? Gram positive. And since this is Word, I don't even think I have to insert a text thing. I think I can just go, I have to turn off this. Let's see. Go back to draw and get rid of the cursor. Good, maybe, and then go back to home. And then now, nope, it's maybe get rid of the cursor now. Oh, there. I just had to go to here where the blank was. Sorry about that. So it's purple, so we know it's going to be gram positive. So you can go. And then we also know that by the shape, it's going to be changed. So that's strepto. I get nervous when I'm videoing, so everything I do is a mess when I video. But there you go, strepto. And then the shape of these are rods. And rods, a rod is bacillus, but plural is bacilli. So streptobacilli, some people pronounce it bacilli. So gram positive for the purple, strepto for the chains, and bacilli, or sometimes I just say streptobacillus bacteria because I'm more comfortable. But that's how you do that. So so I've got this drawn in, and I've got, let's see, gram negative or positive, and then the shape is bacillus, that's rod, and the arrangements is strepto, which is chains. So make sure you pay attention to that. This first one, you can see like little spirals. So you're going to make sure that when you draw it, you're going to go and again, however you can draw. And I do not even see a pink color. Well, this is sort of pink. Okay. And then maybe you're going to make sure you draw some little coilies. And then again, gram negative or positive, and then strepto, is it in chains or not? Staphylo, is it in clumps or not? If it's not in anything, you don't put that. And then you have round is coccus or cocci plural, and then rods is bacillus or bacilli plural, and then sporulum is spiral shape. So you can look at each of these and you can just go up here. If you want bigger circles, you can take this and maybe that's red, and then you can make it really thick there. And look, is there, oh, look, maybe they're in pairs. So find out what the word in pairs means. Make sure you say gram negative or positive. And then if it's strepto, diplo, um, staphylo for clumps, and then there's something specifically for pairs. Okay. And then, so there you go. And then the shape. Okay. So you have bacilli or bacillus for rods. You have round is coccus, and then you have sporulum. So make sure you look at each. Are these clusters? Or they pairs? Are they clusters? Are they pairs? Or are they chains? Are these pairs? And something like this, some of them might look single because when they stir it, it can undo the clumps. But look and see if you see any natural pairs around, and you're not going to stir them together to get a bunch of pairs or chains, but you'll always see when you put them on the side, you do a smear. Okay. But you'll always see, like here, some chains and then some that are broken up a little bit. But that basically is all I wanted to show you with this. If you have any questions, let me know. And let's see, stop share. No, I'll just, yeah, let me know if you have any questions. Good luck with this lab. One other thing. So the plates, the agar plates, they hopefully should have been in your fridge. And there were so far about 2 plates I've heard of that have mold on them already. So if they have mold on them already, they're not going to work. You can do a couple of things. If you're close in the area, I can arrange to get you some more plates. Another thing you can do is you could try to do the lab just with the plates you have, but someone has their split plate is not good. It has mold in it, so that one's not going to work, and that's the clean and dirty hands. So they do have 2 small plates. They could try to draw a line across the bottom of the plate and use one for clean and dirty hands, but they're really small, so it might be hard. It might be a clean and dirty finger, and then it's going to be really hard to breathe over it with the mask. And unmask on 2 sides of a tiny plate, but they can try it. And they draw a line with a Sharpie on the bottom of the plate on the plastic part. Another thing you can do, if you have a plate that's contaminated, let me know. And, I can give you some data for the one of the plate. If all of your plates are contaminated though, then we have to figure out some way to get you some plates. The plates, since we don't have incubators at your house, then they don't just grow in like 2 days like they would if you were at school. But they will grow slower. So they might take 3 or 4 or 5 or 6 days. So, and then there may end up being no growth. So let me know if they seem to be taking too long, but there's been people that they've taken over a week. And that's why this lab, I'm not usually as picky as some others about when you turn them in. But if it's going to be late, let me know it's going to be late. Okay. So that way I'll know that. And I'll still mark it as a 0 whenever I grade them, but then it'll change once you turn it in. Because if I don't put zeros, people get surprised that they're missing something. And I want everyone to know what they're missing and what they need to turn in. And I guess that's it. Have a great weekend. Bye. And email me if you have any questions.